So today's lecture is going to be on aging and skeletal muscle metabolism. First off, and as always, a couple quick announcements. So today was the last chance to view your midterm. If you need to make alternate arrangements with me, that's fine. I can arrange to meet with you and we can look it over. All the papers for presentations are now posted on Sakai, so up until environmental stress is our last topic. Please start thinking about case study number 2. It's due not this Friday, but the following Friday, March 20th. And the thing we talked about in class today is the idea that old papers are still good papers. So if the paper is from 1975, it means you can still use it. If the paper is from 2020, it's still good as well. An update on our final exam. It's Monday, April 13th, 7 to 9 p.m. I apologize. I didn't have any say in picking this, but I also noticed it's the day of the 4P10 exam as well, so apologies to you guys in advance. Today's lecture, same as always, we'll start with an introduction to the topic, so an introduction to aging. We'll talk about insulin sensitivity before moving into our kind of common theme of lipid, carbohydrates, and proteins. We'll then take on a few other angles like menopause, exercise training, and caloric restriction. So of course, our introduction to aging, we all know that Canada's population is aging. A couple statistics to kind of jive with this is the idea that in 2014, over 6 million Canadians were over the age of 65, so it's about 16% of the population. But statistics show that by 2030, over 9.5 million Canadians will be over the age of 65. So a rise from 16% of the population to 23% of the population. We discussed in class some of the ideas of some of these influential factors. So on the left side of the figure, we can look at the idea of an increased population. So the population in thousands is rising, but the population of older adults then is obviously rising. But we can also think about this in terms of a life expectancy kind of phenomenon. We have better healthcare systems, better exercise and longevity systems in place. So if we look at the right side of the figure, we see that the percentage of the total population over 65 is also increasing. So those baby boomers are now kind of entering that 65 plus category. Really, all of these things highlight the important avenue on why we need to study skeletal muscle as well as skeletal muscle metabolism in relation to aging. So now I just want to cover a couple of kind of whole body phenomena that we see as we go through our years of life. So age and body composition. So in both lean and obese individuals, so on this figure, lean individuals are in the dark dots or dark squares, and obese individuals are in the pink squares. What we see is that the total number of adipocytes, so remember we think of adipocytes, we're thinking of fat cells, we're thinking about body composition, we're thinking about body fat percentage. So as we rise through childhood to adulthood, we see this rise in the total number of adipocytes in lean and obese individuals. This levels off through adulthood, but the thing that we're thinking about is although that we start to see a small decline in our older years, so over 60, it's really this kind of idea we need to think about it in the context of the fact that as we age or as we get up into those older years of life, we're actually decreasing skeletal muscle mass. So it's going to be this relative proportion of fat mass, so our adipocytes, to our fat-free mass, skeletal muscle. And what we see is that the relative amount of body fat actually increases because of how much skeletal muscle mass we're actually losing. Another thing we need to consider is fitness levels as we go through our lives. So what we see here on the left figure is peak VO2, so a measure of aerobic fitness. The gold lines are males and the blue lines are females. And what we see is that as we get older, so up until the age of 30, we see these improvements or these increases in physical fitness. But after the age of 30, we see these declines with advanced aging, or with aging, we see steeper decrements in endurance or aerobic exercise capacity. If we look at the figure on the right, what we're looking at is a percentage change in aerobic capacity or VO2 peak. And again, it's expressed with each decade of life. So if it's above that 0 line, it means we're improving our fitness. So improving our fitness up to the age of 30. After the age of 30 is when we start to see these decrements in aerobic fitness. Again, gold is men, blue is women. And what we see is that actually men appear to have more severe declines in fitness, specifically after the age of 60. So that can be defined by the slope of the curve, where males appear to have a steeper drop-off than females. So now we need to think about some of the contributors to fitness and how that can influence our ability to have aerobic endurance capacity. So again, from the time you're born until 30 years old, fitness status is increasing. We saw that on the last slide, and it's kind of seen as that tipping point on this slide here. At some point in your 30s, you begin to lose both skeletal muscle mass and function. This is something called age-related sarcopenia. We're going to refer to it as sarcopenia throughout the rest of this lecture. So if you took an average adult who was fairly sedentary, physically inactive, you would expect them to lose about 3 to 5% of their skeletal muscle mass per decade of life after the age of 30. Another thing I want to highlight here is that some of the parameters on this graph are different parameters we're going to consider that can contribute to an impairment in aerobic fitness in older individuals. So again, what we want to highlight here is we always think about skeletal muscle specifically as it's the focus of this course, but we always want to acknowledge, we always want to think about the idea that aging, or whatever topic we're talking about, does have whole body effects as well. So how aging affects the entire system. First and foremost, we see a reduction in the respiratory system, so our ability for our lungs to function, bring oxygen into the system, ability basically to deliver that oxygen to our working skeletal muscle. We also see a reduction in cardiac function. A common thing you'll see in older adults is collagen deposition or thickening of the ventricles, basically resulting in reductions in stroke volume, cardiac output, which we know are key contributors to aerobic fitness. Moreover, we see impairments in vascular function, so the delivery of nutrients and the removal of waste products from skeletal muscle is impaired, again through collagen deposition or plaque formation. And of course, for the focus of this course, we're thinking about impairments in skeletal muscle mass or content, as well as skeletal muscle function, so the ability to communicate and use that skeletal muscle to perform whatever action or task that we're thinking about. So just a link to skeletal muscle metabolism here. We're thinking about sarcopenia, where sarcopenia is defined as the progressive loss of skeletal muscle mass and function, or we can consider in lay terms the aging of skeletal muscle fibers. It's a multifaceted phenomenon, so we're thinking about not only structural changes in aged muscle, so physical changes to the properties mechanically of that skeletal muscle, but we're also thinking about diminished regenerative capacity, so the ability for that muscle to grow, to repair. We're going to discuss something called satellite cells later in this lecture. Something we won't touch on too much are neurological changes, so you guys might have talked about in previous courses how the central nervous system and the communication through the brain to skeletal muscle, and how those signals, action potentials might translate to functional movements. And lastly, thinking about those vascular changes where, again, impaired delivery of nutrients or removal of waste products can alter or impair skeletal muscle function. So in class, we talked about this idea of whether it was going to be the chicken or the egg. Is an impairment in skeletal muscle mass and function due to aging itself as a kind of phenomenon, or is it due to the diet and lifestyle or environmental factors that are associated with aging? So whether that is really busy work life that leaves you little time to cook your own meals and exercise, or again, is it this idea that with age, we see these impairments in muscle mass and function regardless of whether we are trained or untrained individuals. And in class, we really came to the conclusion that it can be both. So the textbook definitions that we're really working with here are primary and secondary aging, where primary aging is this kind of inevitable change in cellular function and structure, independent of lifestyle, environmental, or dietary changes, as well as disease. Secondary aging, on the other hand, though, involves changes that interact with environmental influences, lifestyle changes, or disease. Which really leads us to this idea of healthy versus unhealthy aging. So we can really think about primary aging being healthy aging, meaning that in the context of a good diet, good exercise, we still might see changes in skeletal muscle mass, skeletal muscle function. This is considered primary aging. On the other hand, on the other side of the equation, we can consider secondary aging as the context of unhealthy aging, meaning that if we have poor nutrition, sedentary lifestyle, we might have these compounded or maybe different outcomes on skeletal muscle mass and function. The bulk of this lecture is going to focus on unhealthy aging. It's typically in the context of sedentary lifestyle and usually poor dietary habits. In the back half of the lecture, we'll see how healthy aging, so the context of exercise training, might actually play a different role. So the first topic I want to kind of delve into is insulin sensitivity and aging. So what we're looking at here is this idea of the first study provided young and old adult humans with a 75-gram oral glucose tolerance test. So you guys remember that with an oral glucose tolerance test, it's this glucose, 75-gram glucose challenge. People drink it. We measure blood glucose, blood insulin levels over time, and it can be a measure of insulin sensitivity or insulin resistance. So what we actually see is that if we look at the glucose figure on the left, we're looking at blood glucose on the y-axis, time on the x. We see that in the young individuals in the black closed circles, compared to the old adults in the open circles, we see basically that young individuals have a smaller area under the curve in response to the 75-gram oral glucose tolerance test. Interestingly, when we look at the insulin figure on the right, what we actually see, insulin on the y-axis, time on the x. Again, young adults are in the black closed circles, and the older adults are in the open circles on top. We see that young individuals have a smaller insulin spike compared to those older adults. So really, taken together, this suggests that older adults have insulin resistance, or they have less insulin sensitivity compared to young individuals. And the way that we kind of look at this is that in the older adults, we have greater spikes in insulin in response to the same glucose challenge, but an impaired ability to clear that blood glucose. Whereas young adults have a smaller insulin spike, but also a smaller plasma glucose or blood glucose measurement or area under the curve, meaning they use less insulin to clear the same amount of glucose, leaving blood glucose levels lower than their older counterparts. So the other side of the coin, thinking about insulin sensitivity, what this study actually did was they used a euglycemic glucose clamp, meaning they delivered a fixed amount of insulin and they infused it in these individuals. Here they looked at glucose disposal rate. So remember that a higher glucose disposal rate suggests insulin sensitivity. So what we see here is that glucose disposal rate on the y-axis, age in years on the x. The younger an individual is, the higher their glucose disposal rate is. The older an individual is, the lower their glucose disposal rate is. So glucose disposal rate was reduced by 39% in older adults. So meaning with the same amount of insulin present, older adults can clear less glucose. This is significantly prevalent in older adults above the age of 60. So now I want to talk about a stimuli other than insulin, so contraction, which can also mediate glucose uptake. Note that this study is in young adult humans, and what we're looking at here is this idea that acute exercise, so skeletal muscle contraction, can mediate glucose uptake. And what they found was that participants completed an OGTT, or that oral glucose tolerance test, in 3 conditions. Once they arrived after being in a trained state, so they'd been training for months and months leading up to this visit, they then underwent a sedentary lifestyle for 10 days, so no exercise, and they also then performed a single bout of exercise and did another oral glucose tolerance test. And what we can see is that if we're looking at the panel on the left, we're looking at blood glucose concentrations. We see that in response to 10 days of no exercise, there's a significant increase in the area under the curve for that oral glucose tolerance test. Moreover, we see a significant spike or significantly higher spike in plasma insulin levels in response to that oral glucose tolerance test. So again, the spike in insulin without the ability to clear as much glucose, suggesting less insulin sensitivity in these trained individuals who underwent 10 days of no exercise. Moreover, if you look at the glucose levels of the trained individuals, we see that they're better able to clear the blood glucose and use less insulin to do so. Even more interestingly, when these individuals came in after 10 days of no exercise, performed a single bout of exercise, their blood glucose concentrations returned to their trained levels. Although the insulin spike was a little bit higher, they were able to, again, improve their glucose clearance after that 10 days of no exercise. Importantly, and not shown here, is that these improvements in the glucose response can persist for 1 to 2 days following a single bout of exercise in young adults. So the important kind of take-home point from this is really the idea that insulin sensitivity can be manipulated by short-term interventions in young individuals at least. And what we're going to see is that on the next slide, this might be a different story in older adults. So here, looking at a similar kind of undertaking in older adults, they either performed an oral glucose tolerance test in a control state following the morning after a high-intensity interval training, or the morning after a moderate intensity or a endurance exercise task. What they found though, so looking at glucose concentrations on the left, no difference in the blood glucose concentrations, whether it was a control day or whether they exercised, and no difference really in the insulin concentrations except in the high intensity interval group, so those thicker dashes. We see a small rise in insulin, but it didn't really translate to any change in blood glucose. So really what this says is that there appears to be a divergent response between young adults and older adults and their responses to acute exercise in terms of insulin sensitivity. Really kind of honing back to this picture that older adults kind of display this insulin resistance or a lack of insulin sensitivity. So why do they have this impaired insulin sensitivity? So the first thing we're going to look at is impaired mitochondrial function in older adults. So older adults have increased lipid accumulation in skeletal muscle. That's that first box on the left there highlighting intramyocellular lipid content. So we see that Y represents young, O represents old. Older adults have higher intramuscular lipid content. Remember, higher levels of intramuscular fat, in the absence of the ability to oxidize it, can impair insulin signaling through this lipotoxicity effect. And actually, this translates to impairments in mitochondrial function. As we can see in the box on the right, we highlight the idea that mitochondrial TCA flux rate, or the ability to functionally operate the mitochondria, is impaired in older adults. And really the functional outcome of mitochondrial work is to produce ATP, and ATP production from mitochondria is significantly reduced in older adults as well. So increased levels of fat, increased lipotoxicity, impaired mitochondrial function, as well as impaired insulin signaling cascades in these older adults. Another thing we can consider is increased mitochondrial oxidative stress. So remember from the guest lecture on Monday last week that aging can increase mitochondrial derived reactive oxygen species. Remember that reactive oxygen species, or ROS, essentially mean that they can cause damage. So low levels of reactive oxygen species is normal. We have that in our day-to-day lives. But in the context of elevated or increased ROS levels, this is when it can cause cellular damage. So what we see in this schematic on the right side of the PowerPoint is that we have increased ROS production in older muscle, which can cause DNA damage to the mitochondria specifically. If the mitochondria are damaged, they have impaired function. This can lead to more reactive oxygen species generated, more mitochondrial damage, more impairments in mitochondrial function, and this kind of cycle continues as the mitochondria become more and more damaged and less functional. Moreover, we know that ROS emissions can stimulate inflammatory pathways that can impair the insulin signaling cascade. Ultimately, big picture, what we're seeing is impaired mitochondrial function to oxidize fat for fuel. Because of that, we have greater lipotoxicity, or these toxic lipid intermediates. And furthermore, we see enhanced or facilitated impairments of this insulin signaling cascade. So this study here, just an interesting study, what they did was they looked at the influence of inflammation, how it's affected by age, and how it's affected by body weight. So if we're looking at the panels on the left, we're looking at TNF-alpha, which is an inflammatory marker, and we see that young individuals have lower levels of TNF-alpha, older adults have higher levels of TNF-alpha. So more inflammation as you get older. And importantly, TNF-alpha is also related to body weight as well. So they took measurements of TNF-alpha mRNA prior to and following weight reduction surgery in obese individuals. And they found that prior to surgery, TNF-alpha levels were high, and following surgery, there was a significant reduction, which can translate to improved insulin sensitivity. So again, this really just highlights the idea that increased inflammation is another way that we can think about impairments to that insulin signaling cascade in relation to aging. So in the context of lipid metabolism, what I really want to talk about with this aging lecture is specifically mitochondrial function. So what we see here is that mitochondrial content is reduced as we get older. This is true for both trained and untrained older adults. So of course, with exercise training, so the bars on the right side of the figure, we see that individuals who are exercise trained have higher mitochondrial content. That's no surprise. That's something we've seen as a training adaptation in the past. But the new thing here is that comparing the black bars, which is young adults, to the white bars, which is older adults, we see that older adults have significantly lower mitochondrial content compared to young individuals, whether they're untrained or trained. This perhaps suggests an impairment to aerobic metabolism in older individuals compared to the young. So it's not always about mitochondrial content. We need to actually measure functional outcomes of the mitochondria as well. So this study was performed in rodents, and what they found was that mitochondrial ATP provision, a measure of mitochondrial function, was actually reduced in older rats compared to their younger counterparts. So young rats, 12 AD is the marker for young here, light gray bars, have significantly greater mitochondrial ATP content and production compared to older rats, so the 26 AD. This suggests that aged skeletal muscle has impaired function compared to young, healthy muscle. Another measure, if I didn't convince you with the last study, for mitochondrial function is mitochondrial respiration. So we can use a number of different substrates to facilitate ATP production. We can actually measure the response of those mitochondria directly. So this study was performed in humans. They took skeletal muscle biopsies from the vastus lateralis. They isolated the mitochondria and exposed them to a variety of these substrates, whether it is pyruvate malate, glutamate malate, or succinate. And what they found consistently was that younger individuals have higher mitochondrial function or mitochondrial respiration compared to their older counterparts, where older adults have decreased mitochondrial respiration or impaired mitochondrial function. So why might this be occurring? Coming back to our idea of reactive oxygen species, remember, reactive oxygen species in higher amounts cause damage to cellular proteins. So what this study looked at was wild-type or control mice in the black bars compared to SOD2 knockout mice. Note that SOD2 is an antioxidant defense. Antioxidant defenses can reduce the amount of reactive oxygen species present. So what they found was that looking at the top figure with oxidative damage, they compared young animals on the left of this figure to older animals on the right of this figure. And they found, just looking at the black bars first, older animals, with their wild types, had greater oxidative damage compared to young animals. Moreover, when they knocked out this SOD2 gene, they found without that antioxidant defense, oxidative damage also increased, which is perhaps no surprise. But when we go on to look at something like function of the mitochondrial complexes, so that second figure there, we're looking at complex 1 and complex 5, what we actually see is that the wild-type mice, so black bars again, the old mice have impaired or reduced complex 1 and 5 function compared to their younger counterparts. Moreover, the SOD2 knockout mice have impaired function in the young mice compared to the young mice wild type. Because there's no real difference between the SOD2 knockout in the young animal compared to the wild type of the old for complex 1 and 5, we can actually make an assumption or an association that SOD2 function is impaired in aged muscle. What's important about this is that it translates to an impairment in ATP synthesis. So what we see is that, again, looking at the black bar specifically, aged muscle, so aged rodents, have impaired ATP synthesis compared to their wild-type young counterparts. And again, SOD2 knockouts in young animals have reduced ATP synthesis, meaning that there's, again, this link between an oxidative defense system and how it affects mitochondrial ATP synthesis. So something new that we didn't really talk about in the past is this idea of mitochondrial morphology and how it can influence skeletal muscle metabolism. So what mitochondrial morphology is the shape of the mitochondria. And really what we know is that mitochondria are continuously undergoing fusion and fission events in order to maintain proper health and function of those mitochondria. Where fusion is really where we take one or two or more mitochondria, fuse them together to create one bigger mitochondria, their contents mix. Fission is when we actually take those mitochondria and split them back apart to form new, functional, regenerated, renewed mitochondria. What this study looked at was a marker of mitochondrial fusion, so MFN2, as well as a marker of mitochondria fission, DROP1. And what they found is that the ratio of fusion to fission is significantly higher in aged skeletal muscle. If we think about that, break it down and put it into context, what that really means is that there's more fusion, so more mitochondria are actually linking together to create bigger mitochondria, they're mixing, but there's less fission to split them apart. What this actually does is it creates large, so because there's two that have linked together, they've joined all their contents, it's bigger, large, but also misshapen mitochondria, and because they're misshapen and larger, they have impaired function. So that's how we actually get decreased mitochondrial content because they're fusing together, and we get decreased mitochondrial function because they're fused together, which actually can impair their ability to work properly. So that's what I wanted to talk about on the lipid metabolism front, specifically related to mitochondrial function. Now I want to go on, change gears, think about a few elements of carbohydrate metabolism that can influence or can be influenced by aging. So here we're thinking about some key glycolytic enzymes. First, we'll talk about hexokinase. So we know hexokinase converts glucose to G6P. Remember, G6P, once it's formed, it traps that glucose inside skeletal muscle, so it can't leave again. What we see is that in the top figure here, hexokinase levels are higher in younger individuals and lower in older adults. Same side of the story, but different enzyme, lactate dehydrogenase, which catalyzes the reaction of lactate to pyruvate or pyruvate to lactate, either way, higher in younger adults, lower in older adults. And what we see is that this may have the capacity to translate to impaired glucose metabolism. Something we could consider is that it may be a contributor to fatigue during exercise. So now I want to look again at more of these glycolytic enzymes, thinking about the muscle fiber type specifically. So in class, we talked about this idea that this figure here shows us soleus in aged versus young rodents. Not a whole lot of difference in glycolytic enzyme activity, but perhaps that's not surprising based off of the muscle type that preferentially uses carbohydrate metabolism, and that would be our glycolytic muscles. So plantaris is a glycolytic muscle, and what we see here is that young rodents have higher levels of these glycolytic enzymes compared to their aged counterparts, which have lower levels of these glycolytic enzymes. Perhaps not surprising again, because the plantaris is a glycolytic muscle, it's likely that we would see any changes in a muscle that would predominantly rely on carbohydrate metabolism. Here we're looking at glycogen synthase activity. Remember, glycogen synthase promotes the storage of glycogen through the addition of glucose molecules to the existing glycogen molecule. So what we see is that the top figure is showing us older rodents, middle-aged rodents in the middle, and at the bottom would be older rodents. Sorry, so the top is young, the middle is middle-aged, and the bottom is older rodents. So basically what we see is that the ability to synthesize glycogen via glycogen synthase is impaired with aging. So that bottom line is going to be our old group. They have less glycogen synthase activity, meaning that they may have an impaired ability to synthesize and store muscle glycogen. The last kind of picture I want to talk about on carbohydrates is this idea of glycogen utilization during exercise. So what we're looking at here is rodents in response to electrical stimulation, so skeletal muscle contraction, so forced kind of exercise. This figure is looking at glycogen depletion, so lower levels means more glycogen has been depleted. If there's more remaining, it means less glycogen has been depleted. So what we see here is that young rodents utilize more skeletal muscle glycogen than old rodents. So the 28 months is the old, 9 months is the young. Old group does not use that much skeletal muscle glycogen compared to the young animals. What's important about this is that these authors actually standardize their results for the fact that they know that older animals have less skeletal muscle mass, so it makes sense that they'd be able to store less glycogen. But despite that, they're looking at glycogen depletion or glycogen breakdown and utilization per gram of muscle, meaning that they normalized for skeletal muscle mass, and still, older adults have impaired abilities to use skeletal muscle glycogen compared to the young animals. Maybe this contributes to impairments to athletic performance in older animals, possibly linking again back to this idea of earlier fatigue onset. So now we're going to shift gears thinking about protein metabolism. So what this figure really told us was looking at age on the X, muscle strength on the Y. What we see is that, thinking back to an earlier figure from this lecture, around early adulthood, 30 years old, we see this peak in muscle strength. So what we want to do in early adulthood and in our youth is to maximize skeletal muscle strength. Because in our older adult life, or adult life, our goal is to maintain skeletal muscle mass. And in our kind of 60 plus older life, we want to minimize skeletal mass losses. This is because it becomes more difficult to build skeletal muscle mass in older age. So really, again, early life we want to maximize so that in adult life we can maintain, and older life we can minimize our losses, again, of skeletal muscle mass and function. So what this first study looked at was muscle protein synthesis. We know our two main contributors for skeletal muscle protein turnover are going to be muscle protein synthesis and muscle protein breakdown. So this study looked at 42 healthy volunteers, young, middle, old, good mix of males and females. At rest, they delivered 13 carbon-labeled leucine, radio-labeled leucine in order to measure muscle protein synthesis. What they found is that resting muscle protein synthesis is reduced in middle age and older adults compared to young. Again, contributing to this idea that we want to maintain skeletal muscle mass in middle adult age and prevent our losses in older age. But we need to consider whether this muscle protein synthesis scenario is true for all scenarios. So what about the context of anabolic stimuli, whether that is amino acids or resistance exercise? So we need to consider something called anabolic resistance in older adults. So in this case, our anabolic stimuli is going to be the delivery of amino acids. This study looked at 44 young and elderly men. What they did was they measured muscle protein synthesis, again by this incorporation of radio-labeled leucine into the muscle. They provided a dose-response curve of different protein doses, mixed essential amino acids. What they found, and what we've seen in the past with other protein lectures, is the idea that 20 grams of protein should maximally stimulate muscle protein synthesis. But what they actually did was they, in the older adults, delivered a final dose of 40 grams of protein just to see if it would tip the scales to bring the older adults up to the level of the young adults. Unfortunately, what they found is this idea of anabolic resistance in older adults. Despite the same levels of protein delivered, whether that was 0, 10, 20, or 40 grams of protein, the amount of muscle protein synthesis was significantly lower in those older adults compared to the young individuals, again, termed anabolic resistance. The other idea of anabolic resistance that we're going to talk about is the other anabolic stimuli, acute resistance exercise. So it should increase muscle protein synthesis. They took 25 young, 25 older men. They looked at, again, this incorporation of 13C radio-labeled leucine into skeletal muscle. And what they did this time was they did a dose-response relationship between muscle protein synthesis and exercise intensity. So the first figure here is looking at increasing exercise intensities as a percentage of 1 rep maximum, where increasing intensity should increase the muscle protein synthesis expressed as this fractional synthetic rate. They see that in older adults, again, there is a lower fractional synthetic rate in response to these percentage of 1 rep max compared to their younger counterparts. If we look at the figure on the bottom, they also followed muscle protein synthesis in the 0 to 4 hour period following exercise in these individuals in response to a meal. They found that muscle protein synthesis in both groups peaked around 2 hours. But again, older adults had an impaired muscle protein synthesis response post-exercise compared to their younger counterparts. Again, highlighting this idea of anabolic resistance in older adults. But we know that muscle protein synthesis isn't the only side of the equation. We also need to consider muscle protein breakdown. So what this study actually looked at was 8 young and 8 older men. They looked at interstitial 3-methylhistidine, or 3-MH. This is a proxy for muscle protein breakdown, where higher 3-MH represents greater muscle protein breakdown. Just looking at any of the figures on the right side of the diagram here, we're looking at the idea that muscle protein breakdown is greater in older men compared to younger men, so the gray bars versus the white bars. And they measured this at 2 different sites, so probe A and probe B, and they also took a combined average, and the story was the same regardless of the site they used. So if we take this in stride and we think about this in the context of protein turnover, what we actually see is that older adults are more likely to be in negative protein balance because they have lower muscle protein synthesis in combination with greater muscle protein breakdown. So another new topic that we haven't talked much about in the past is the idea of satellite cells. So satellite cells are stem cells found in skeletal muscle, and really their goal is to allow for skeletal muscle growth, regeneration, and repair. Satellite cells are activated in response to muscle injury or stress. So we talked about the idea that skeletal muscle contraction during resistance exercise is creating these micro tears in the muscle. That's an example of muscle injury or muscle stress. What's interesting is that some of these activated satellite cells, so they're activated by skeletal muscle contraction, for example, they can actually self-renew and return to an inactive state. What's cool about this is that it creates and maintains this satellite cell pool that can be called upon, activated, inactivated, activated, inactivated in response to the stimuli that it sees. Interestingly, when we have impairments in this satellite cell cycling, it's been correlated or been linked to insufficient muscle repair regeneration, which can actually contribute to atrophy or age-related impairments in skeletal muscle function, which we're going to see in the coming slides. So aging in satellite cells is a complicated topic. I just want to give you guys a glimpse into some of the avenues that are influenced by aging in terms of satellite cells itself. So aged muscle is shown to possess a diminished or delayed regeneration of these satellite cells. Or aged muscle, is shown to possess a diminished or delayed regeneration. So skeletal muscle actually turns over more slowly, regenerates, repairs more slowly. You might hear about older adults who have an injury that, back in the day would have only taken a couple of days to heal, but now suddenly it's taking weeks to months. That's kind of this link here. What we tie this back to satellite cells then is the idea that older adults have a reduced number and impaired functioning of those satellite cells. So there might be this miscommunication within that satellite cell pool where this activation, inactivation, and the regeneration of that satellite cell pool isn't as healthy as in young adults. Really taken together, this means that older adults have an impaired ability for muscle growth, regeneration, which can contribute to this idea of anabolic resistance or even long-term injury. So now switching gears, thinking about menopause and how that can influence skeletal muscle metabolism. First thing I want to highlight is what exactly menopause is. It's a period in a woman's life around 45 to 50 years old on average. It can occur much earlier, it can occur later, but again, 45 to 50 is an average. What it's marked by is the cessation or the end of menstruation for 12 consecutive months. In that 12-month period leading up to official menopause, we call this the menopausal transition or perimenopause. And really what's important in our kind of view for menopause is the idea that the ovaries decrease and then eventually stop producing female sex hormones, estrogen and progesterone. And in the kind of context of this course, we really focused on estrogen and will continue to do that in this topic as well. So we know that estrogen can regulate skeletal muscle metabolism, where estrogen can bind to its receptor. It can elicit changes in signaling as well as protein content and mRNA. But what's more important here is this link between estrogen action and metabolic health in women. So there's a lot of literature out there that suggests older women who are menopausal have this sudden increased risk in type 2 diabetes, obesity, cardiac disease, and really a lot of them undergo exogenous hormone replacement therapy as a tool to mitigate some of these disease outcomes, where it's been shown that estrogen or progesterone or the combination of estrogen and progesterone therapy can actually decrease the risk of metabolic disease, whether that's type 2 diabetes, obesity. It can even improve cardiac function and overall health. So now I want to talk about estrogen in the context of insulin resistance. So this first study is just to highlight the key role that estrogen plays and how it actually might have a role between men and women in terms of insulin sensitivity. So this is a study in young rats, and what they found was that they looked at the basically infusion of excess lipids. So we know excess lipids can cause insulin signaling problems or insulin resistance because of these toxic lipid intermediates that can interfere with the insulin signaling cascade. What they found was that when they provided these animals with a lipid infusion during a euglycemic hyperinsulinemic clamp, and they looked at insulin signaling cascade function in males who had an increase in plasma-free fatty acid, it resulted in about a 40% reduction in insulin-stimulated glucose uptake, so glucose infusion rate's the top figure, and this was also linked to impairments in phosphorylation of IRS1. So remember, in this case, phosphorylation of IRS1 is making it active normally. So if we get less phosphorylation of it to its active form, there's impairments to that insulin signaling cascade, less glucose infused because less glucose is uptaken. However, when we look at the female data, we see in response to this lipid infusion, no impairments in glucose disposal rate, so no impairments in glucose uptake, and no impairments to the insulin signaling cascade, specifically at IRS1, which is really interesting. It suggests that estrogen may be protective against insulin resistance in the context of lipid overload. When we take this to the next step, really the next study in a logical progression would be to take young rats that are overreactimized, so OVX, meaning they don't have any circulating sex hormones, and taking those overreactimized rats and providing them back with estrogen to see if estrogen is kind of this causative factor. Again, they assessed insulin sensitivity via euglycemic hyperinsulinemic clamps. Remember, a higher glucose infusion rate suggests insulin sensitivity, meaning they're able to clear that glucose challenge. What they found was that compared to young animals, these OVX rats had impaired glucose infusion, so insulin resistance. And when they provided these OVX rats with estrogen, it restored their insulin sensitivity and increased that glucose infusion rate, which is really interesting, suggesting that estrogen plays a key role in insulin sensitivity. Same thing on this B panel figure, we see whole body glucose uptake impaired in these overreactimized animals, provide them back with estrogen, and they're restored to the young healthy state. Again, highlighting this key role for estrogen in insulin sensitivity. Next, we need to think about the role estrogen plays in mitigating lipotoxicity. So this rodent study utilized control versus estrogen receptor knockout mice, so the MERK-O mice. What they found, looking at the panels from left to right, was that these MERK-O rodents had increased inflammation measured by JUNC as well as I-kappa-kappa beta, two measures of inflammatory status, higher in these estrogen receptor knockout animals, which also correlated with higher accumulation of toxic lipid intermediates, so DAGs and ceramides. Ultimately, this resulted in impairments to the insulin signaling cascade and ultimately insulin resistance, meaning that our estrogen receptor knockout mice had impaired responses, so phosphorylation of IRS1, in response to insulin, meaning they had impaired insulin signaling cascade function, aka insulin resistance. Again, showing us that estrogen and its interaction with its receptor is key for metabolic health and insulin sensitivity in female animals. Again, changing gears, thinking about exercise training now. What I want to show you here is that, unfortunately, the reality of aging is that even if you're fit and trained, there are natural decrements in performance associated with aging. So these are male master sport performance measurements. So masters athletes are just older individuals who are competing at a high level. What they see is that across all of the training sport modalities that we look at, there are natural decrements in performance as they go from 30 years old up until end of life. Now, it's not all doom and gloom. Other things that we need to think about are from a functional standpoint. This figure is looking at aerobic capacity, so VO2 peak on the y-axis and the age on the x. And what it's showing us is that the slope or the rate of decline is not different whether you're trained or sedentary. So you lose about 10% of your aerobic capacity with each decade after 30 years old, which is scary. But it's an advocacy for improving your physical fitness and health in your early years so that you can maintain a higher VO2 max or higher fitness level for longer. If you start at a higher level and your rate of decline is 10%, you could be at the level a sedentary person is when you're 70 compared to 40. Really what this means in the bigger picture is that it could translate to greater independence for longer, so kind of staying out of that old folks' home. But really what we need to think about in terms of adults or older adults is not really aerobic fitness for the most part. It may be less important, and we may think about strength being the more important factor here. So we need to think about strength in terms of carrying your groceries, doing gardening, household chores, functional and practical parameters, which is why most of the research in older adults really focuses on strength training for this reason. So here, plain and simple, what the study is showing us is that they recruited males 60 to 70 years old. They put them through a 12-week training program with 3 sessions per week. All I want to show you here is that their strength significantly increased in both the knee extensors and the knee flexors. Big picture is that there's no doubt that older adults can respond to a strength training program. So even though they might have anabolic resistance, lesser increases in strength, it doesn't mean they won't have improvements. In fact, they will. They may just be to a lesser degree than you would see in a young individual. So when we look at these improvements in these adults following a strength training program, they actually looked at muscle hypertrophy, as well as the number of muscle fibers and their composition prior to 6 weeks into this training program and following the 12th week. What they found was that in panel A, significant increases in skeletal muscle hypertrophy in both the right and left legs. They also found in panel B, significant increases in the number of skeletal muscle fibers for type 1s, type 2s, and MFA is mixed muscle fibers. Meaning, taken together, older adults following 12 weeks of resistance training can have increased cross-sectional area of the muscle, so hypertrophy, as well as an increase in oxidative and glycolytic fibers, or otherwise defined as an increased number of muscle fibers altogether. The important thing to note, though, is thinking back to anabolic resistance, this response is going to be lower than that seen in younger individuals. Coming back to this figure here, the key point I want to highlight is when we're considering strength training in older adults, and because they have anabolic resistance, maybe a nutrition recommendation could be that the elderly or older adults may need to increase their protein consumption to kind of offset this anabolic resistance. So now I want to talk about different training modalities and whether one is better than the other to promote hypertrophy, muscle protein synthesis in older adults. So what this study did was it compared low 40% of 1 rep max and high 80% of 1 rep max intensity resistance exercise in older adults. They performed basically the same amount of work in both groups, and they just wanted to know if low load, high reps versus high load, low reps made a difference in terms of the gains in strength that older adults saw. What they found was that there's improvements in both groups, obviously, with the strength training program, but the greater improvement was actually seen in the high resistance, low repetition group. In class, we discussed the idea of practicality, so One angle that was raised was the idea of loading the skeletal muscle to promote bone strength. So that's a great angle for the favor of 80% 1 rep max. Another angle we could consider may be the safety angle, in which frailty may play a role in what kind of training modality or training program you could suscept your older adult population to. So again, just things to think about, ideas, and taking this outside of the context of strictly learning in the classroom, so thinking about the bigger picture. The last idea I want to talk about in terms of exercise training is going to be diet versus exercise in older adults. Is one better than the other? Is the combination the best? This study looked at obese older adults and the effects of diet alone, exercise alone, or the combination on weight and basically quality of life. So these people were 70 years old, overweight, 37 kg per meter squared BMI. For the diet intervention, it was a reduction of about 500 to 750 kcals per day, and the exercise was 90 minutes per week, which is less than the typical recommendation for obviously young, healthy individuals. But these obese older adults, that was definitely an exercise program that they were susceptible to. Looking at this figure here, whether it was exercise, diet, or the combination, compared to the control group, significant and similar reductions in body weight with each protocol. So in terms of a weight change parameter, they're all the same. But what we need to think about now is this idea of aerobic capacity and quality of life. What they found consistently was that diet and exercise in combination was more effective than diet or exercise alone. So this makes sense because if we think about diet and exercise in combination, we have both a reduction in body weight as well as an improvement in fitness. And what we do is we typically express VO2 peak relative to body mass. So if we reduce our body mass and improve our physical fitness, it's most likely we're going to see an improvement in VO2 peak. More importantly, perhaps, we see a significant improvement in the quality of life of these individuals who are on a good diet and have proper exercise. Seems logical as well. You eat better, you exercise more, you probably feel better than each of them alone. So the last topic I want to go on about is aging and caloric restriction. I think this might be a new topic for a lot of you, and something that is actually really up and coming and interesting. So caloric restriction, although I think of it like eating a single lettuce leaf, what it really is reducing your average daily caloric intake below what is typical or habitual. Importantly though, we're not doing this in the context of malnutrition. We're always having the adequate nutritional profile of a healthy individual, so we're meeting our macro and micronutrient requirements. Typically, when we see caloric restriction studies in rodents, it's pretty aggressive. It's about 40% reduction. So if you wanted to compare this to a human diet, it would be like taking a 2,000 kilocalorie diet and dropping it down to 1,200 calories per day. Basically cutting out an entire meal. I know that wouldn't make me very happy. So typically seeing human studies looking at caloric restriction, it's typically more modest, so 10 to 15% reduction. I think the largest I've ever seen in a study was 35%, and they probably had quite high dropout rates in that study too. But what's really cool about caloric restriction and its use in scientific context and even lifestyle context is that it's the only intervention known to date that consistently decreases the biological rate of aging and increases both average and maximal lifespan. It's a pretty bold statement, but I'm hoping I can show you some of the literature to kind of back up that claim. So first, looking at a rodent study, so they were placed on a normal diet, so AL just means ad libitum, so they can eat when they want, or a diet that had 40% reduction in calories, so caloric restriction or CR. What we see is that in the young animals, so 8 to 10 months old, that's considered young, there's no difference in mitochondrial respiration between either of the ad libitum or calorie restricted animals. But what we actually do see is that in the ad libitum group at 35 months, so the old rodents, mitochondrial respiration is significantly reduced compared to the young animals. More interestingly, if we look at the older animals in the calorie-restricted group, so they're calorie-restricted their whole life, you see that they have impaired or preserved mitochondrial function similar to the young rodents. Really taken together, big picture, caloric restriction may prevent age-associated declines in mitochondrial function in rat skeletal muscle. So if you're put on a calorie-restricted diet at a young age, you might be able to stave off some of these mitochondrial function impairments commonly associated with older age. So why might this be happening? Something that another group of researchers sought to investigate was this idea of aging and oxidative stress. So what they did was they took young rats and they were placing them on either a normal ad libitum diet or a calorie-restricted CR diet. What they found was that looking at oxidative stress over the lifespan of these animals, animals who are on a calorie-restricted diet had significantly less oxidative stress as they aged compared to the ad libitum normal diet. So if we look at the panel on the left, SH is just a thiol group. Thiols are targets for oxidative damage. So if you have more thiols that are intact, it means you have less oxidative damage. So what we see is that the calorie-restricted group has greater number of thiols intact, less oxidative damage. On the other hand, carbonylation is a measure of oxidative damage. If a protein becomes carbonylated, it has been damaged. So the ad libitum group has more oxidative damage because it has higher carbonylation compared to the calorie-restricted group, which has less carbonylation, less oxidative damage. So why might there be more oxidative stress? This study sought to investigate that question, really wanted to understand where this problem is coming from. Remember, antioxidants are able to battle oxidative stress or reduce the number of reactive oxygen species. Reactive oxygen species are what cause that oxidative stress, cause that damage to those proteins. If we look at the figure on the bottom here, what we see is that rats on a normal diet have increased ROS production. This is measured by a free radical called superoxide. Superoxide is a reactive oxygen species and can cause the damage to proteins of interest. So again, young animals ad libitum have the same amount of oxidative damage as calorie-restricted older animals, whereas ad libitum animals, so normal fed animals who are older, have higher oxidative stress as measured by superoxide. Why might these animals have more oxidative stress? So rats on a calorie-restricted diet actually have antioxidant defense systems that are similar to young ad libitum animals, whereas animals who are on an ad libitum diet and are aged actually have reductions in these antioxidant defenses. And that's seen on the right-hand side here, looking at ABC. We see these ad libitum older animals have reductions in SOD, catalase, and glutathione, which are all antioxidant systems in our body. This really led us to the question of whether a calorie-restricted diet would be feasible for humans. There's a couple angles that we discussed in class. First, a lot of these calorie-restricted diets are actually performed from a very young age in these animals. So they start at an age that's comparable to starting a calorie-restricted diet in a 10-year-old. And really, from another feasibility standpoint, could you reduce your caloric intake from 2,000 calories to 1,200 calories happily for the rest of your life? So what this last slide is really highlighting, kind of the take-home picture that we're looking at, is this idea that rats were on a normal diet or a calorie-restricted diet at the beginning of their life. After 4 months, half of the animals were actually switched to the opposite diet. So if they were in the ad libitum group, some of them stuck with it for the entire duration of the study. Others went from ad libitum to calorie restricted. Others were calorie restricted for their whole life. And others went from calorie restriction to ad libitum after this transition period. What these authors really wanted to do was to determine whether starting a calorie-restricted diet later in life could produce the same effects as starting it at a young age. So maybe making this more translational to humans, could we start a calorie-restricted diet in our 30s or our 40s and expect to have a longer life, a healthier life with less complications in terms of oxidative stress or mitochondrial impairments? Unfortunately, what they found, really focusing on this group that was started with an ad libitum diet and then switched to calorie restriction, they found that once the oxidative damage had occurred in these animals, so carbonylation or T-bars, which is another measure of oxidative damage, they found that once that oxidative damage had occurred, unfortunately, it was not possible to reverse it with 6 weeks of a calorie-restricted diet, meaning that possibly it wasn't a long enough duration, but really meaning that maybe it's not so feasible for humans who could be practicing this calorie-restricted diet once they're more informed around the age of 30 or 40. With that, on Thursday, we will have the appetite hormone presentations, as well as next week, Monday, we switch kind of topics moving towards ergogenic aids and athletic performance.